Assalamualaikum and hi, my name is Muhammad Khairi bin Mazaki. My metric number is S48184 and today I would like to give some short briefing about my thesis presentation. Okay, my topic is the career preservation of Artemia franciscana in liquid nitrogen and its effect on the Artemia viability. Okay, first of all is the Artemia, also known as a branch shrimp, which is come from the class of Crustacea and from the order of Anastrika. So, the main important process in this experiment is career preservation, which is use of very low temperatures to preserve structurally intact living cells and tissues. Okay, next is liquid nitrogen is used in this experiment because it has an extremely low temperature. At the end of this experiment, we can see that the Artemia can be survived or not. Okay, for the introduction is, the Artemia are currently the most commonly used life level feed organism in the aquaculture industry because of the various life stages and it contain high nutritional value for the larvae. During adverse conditions, the Artemia will produce resting egg called cysts that can be stored for a long time. So, the cysts will be hatched when the environmental condition is optimum. Okay, what is career preservation? As I explained before, the career preservation is the process of using a very low temperature to keep the cell and tissue intact. The problem statement. First, long culture period is needed for the Artemia to reach the adult size. So, as we know, the Artemia need 14 days to reach the adult size. So, by using this method, the Artemia can be used immediately for feeding to the fish larvae. Then, secondly, the viability of Artemia franciscana after career preserve is unknown. So, the effectiveness of career preservation method can be evaluated to determine the viability of career preserve thawed Artemia snowplant. The objective of this experiment is to investigate the survival and the viability of Artemia snowplant after career preservation process. Okay, this is the experimental design. First, 3 grams of Artemia cyst was weighed and placed in an aquarium filled with 5 liters of seawater and strong aeration for 24 hours. After 24 hours, the Artemia was hatched and the Artemia nopli was siphoned and harvested with a 200 micron mesh prior to use in this experiment. Okay, briefly, 10 nopli respectively was prepared and placed in 1.5 ml of cryovice. The Artemia's nopli was triplicate, so the total of 30 of Artemia was used for each experiment. By using the forceps, the vials was dipped into liquid nitrogen and stored for 10 minutes. After 10 minutes, the cryovice was exposed to room temperature for 30 seconds and then immersed in water of 28 degrees Celsius for 1 minute. After that, the sample was placed in a beaker of 100 ml containing 80 ml of pre-aerated seawater of 28 ppt. After that, the viability of the nopli was observed after being thawed. Okay, this is the statistical analysis. The survival of Artemia's nopli was determined every 8 hours for 24 hours. So, the survival was calculated by using the formula of the survival below. Okay, for the result, all the Artemia nopli cryopreserved in liquid nitrogen could not be revived after thawing at room temperature. Total mortality was observed. This is the Artemia nopli before the cryopreserve, and this is the Artemia nopli after being cryopreserved. Okay, for the conclusion, the process of cryopreservation has caused none of the Artemia nopli to revive. They could not survive after being freeze-thawed. This is due to seawater in the cryovial that form ice crystal on nopli, which has also penetrated into their cell as well. Okay, suggestion for further study is, first, maybe we can add a cryoprotectant to avoid ice crystal formation during the cryopreservation process. We have to know the suitable amount of extender to be used during this process. And we have to study the other methods that are suitable to cryopreserve the Artemia. Okay, this is my Gantt chart, which is 100% complete. Okay, this is my references. Okay, that's all from me. Thank you.